Hi, everyone. In this short talk today, I'm going to provide some highlights of the roadmap to naming uncultivated Archaea and Bacteria paper that was published in Nature Microbiology in June of last year. Also going to provide some updates on where we're at now. This effort was initiated by Brian Hedlund and Anne-Louise Reisenbach in a program supported by the National Science Foundation. This program was called Microbial Systematics for the Next Decade. This included virtual webinars, which provided background on current proposals to revise microbial systematics. And then there were two face-to-face workshops. The first focused on creating a unified classification system for cultivated and uncultivated Archaea and Bacteria. And the second focused on addressing microbial taxonomy, database, and bioinformatic challenges to incorporating uncultivated microorganisms. The goals were to entrain diverse expertise from stakeholders in microbial systematics, microbial ecology, evolutionary biology, and eukaryotic systematics. The effort also created a platform for scientific community discussion and consensus building, and provided a forum for education of principles of nomenclature according to the ICMP. We also took an in-depth look at the challenges posed to a roadblock in nomenclature of majority of Archaea and bacterial diversity. The last goal was to develop a cohesive plan to integrate uncultivated and fastidious bacteria and Archaea into a systematics framework. The problem that we're facing is that of sheer magnitude of microbial diversity. A growing proportion, estimated 40% of archaeal and bacterial diversity, is solely represented by genome sequences. Uncultivated Archaea and bacteria are not formally recognized in the ICSP nomenclatural system. And this field has become isolated from mainstream microbiology and has generally been resistant to innovation. The basis of this conflict is that current standards based on strain phenotype characterization are at odds with genome mapping, which enables communicating the extent of microbial diversity on Earth. These issues have been discussed in the literature in the last several years. A proposal was put forward by Barney Whitman in 2015 that proposed type material for taxonomic descriptions to be genome sequences. There were several other papers, including one by Konstantinidis et al. In 2017, that suggested that microbes that are uncultivated are in need of their own taxonomy. Further papers have addressed this, including a paper by Donovan Parks suggesting a phylogenomic basis to revise taxonomy. There are a few terms I wanted to just clarify as they're going to be important going forward through this workshop. Systematics and taxonomy are synonymous terms, and they circumscribe both nomenclature and classification. Nomenclature, which is the focus of this workshop, is a system for giving names to organisms where the primary purpose of giving a name is to supply the means of referring to it or communicating it. Classification is the placement of names into groups on the basis of evolutionary relationships, as well as similarities and shared qualities or characteristics. Nomenclature is overseen by the International Committee of Systematics of Prokaryotes. This is the body that supervises the publication of the ICMP, which is the code, and whose judicial commission issues opinions concerning nomenclatural matters and revisions to the code. The ICMP, which is referred to as the code, is the definitive set of rules, principles, and recommendations for naming bacteria and archaea. The code requires that every name is associated to a nomenclatural type. All names are announced. Names that are validated first have priority. Priority, in this case, is the first validly published name that is correct and supersedes non-valid names. Currently, taxon names require the etymology of the name, taxon description, designation of type material, deposition of pure cultures into 2 international culture collections in 2 different countries. Where we are now is that there are over 700 Candidatus species that have been formally proposed in the literature. However, many of these are incompatible with the code. There are many alphanumeric names representing various taxonomic ranks, such as phylum, class, order, that do not have reference material. There is also a large degree of synonymy amongst the classified taxa, which causes challenges in the literature. There's also, as we know, a flood of new taxa being based on MAGs and SAGs that have been published. One study found that nearly 16,000 of nearly 25,000 proposed species are solely represented by DNA sequence. Thus, in the roadmap manuscript, we put forward two different plans. Plan A, which is to modify the existing code, and Plan B, which would be overseen by an international society and would establish a code for uncultivated organisms and their nomenclature. There is a commonality between both plans, and that both would need to have ad hoc committees that would be tasked with evaluating the extant Candidatus names, resulting in an approved lists 2.0. That they would also determine the persistence of using Candidatus versus other designations for uncultivated microorganisms, such as a superscript. They would minimize redundancies with other taxonomies, publish validation lists for uncultivated taxa, and establish minimum SAG and MAG quality standards. The two then deviate, and that the one on path on Plan A would modify the existing code in which DNA would be approved as type material. DNA-based nomenclature would have standing and priority. New names of uncultivated taxa would appear in validation lists. And under Plan B, the new code, an uncultivated code, would create DNA-based classification for uncultivated microorganisms that would follow rules that are set forward, and new uncultivated taxa names would appear in digitally accessible lists or databases. Plan B has been conceived as a parallel of Plan A, such that they could be merged in the future. Sequence-based nomenclature requires next-generation tools. Guidelines for genome standards are required, including metadata, such as contextual information, minimal standards, including completeness and contamination, and designation of type material. Tools for digitization of prokaryotic nomenclature are also required, and the system would be amenable to automated naming, creating digital protologs. Valid nomenclature lists could be provided based on sequence information, data, and a system for crosstalk amongst databases could be established. There are a few take-home points that I would like to make. The first is that microbial cultivation is the gold standard required to study the phenotype and physiological characteristics of Archaea and Bacteria. In no way is a sequence-based nomenclature intended to dissuade cultivation, nor impede the growing momentum in cultivation innovations that have been occurring in recent years. Naming organisms based on sequence as type addresses a dire need in the community to be able to communicate diversity of life that is recognized as a result of technology achievements across microbiology. In March 2020, the ICSP voted on the Whitman 2015 proposal to use gene sequence data as type. There was a community-wide discussion that was online in February and in March, and then at the end of March, the votes were in. The result was that the proposal was rejected, and this is summarized in a paper by Sutcliffe et al. In 2020. So we are now moving forward with Roadmap Plan B. The talk that follows mine by Barney Whitman addresses this. I would really like to acknowledge the attendees of the Microbial Systematics for the Next Decade workshop, the co-authors of the manuscript, and those that endorsed it. Thanks for your time.